Dear viewers, greetings. In this video, we are going to see about an autoclave. The contents of this video includes an introduction about autoclave, principle of autoclave, types of autoclave, instrumentation of autoclave, steps for using autoclave, applications of autoclave, advantages of autoclave, limitations of autoclave, precautions for using autoclave. Autoclave. An autoclave is a pressure chamber used to carry out the sterilization under pressure using moist heat. An autoclave is a machine that provides a physical method of sterilization by killing bacteria, viruses, and even spores present in the material put inside of the vessel using steam under pressure. Charles Chamberlain invented the modern autoclave in 1879. He worked under Louis Pasteur and created this device to support Pasteur's studies in microbiology. Autoclave is used to sterilize the dressings, instruments, laboratory ware, culture media, and pharmaceutical products. Principle of autoclave. The mechanism, design, and principle of the autoclave is similar to the pressure cooker. The autoclave works on the principle of moist heat sterilization. Autoclaves typically yield a temperature of about 121 degrees Celsius, taking about 15 to 20 minutes to complete the sterilization process. However, autoclave cycle may be adjusted accordingly by the working technician. The high pressure inside the chamber increases the boiling point of water for the sterilization of equipment while ensuring the rapid penetration of heat into the deeper part of equipments. Steam sterilization using autoclave is effective because the moisture of the steam helps to coagulate the proteins that microbes thrive on. Coagulating the proteins disables the microbes and eventually kills them. Types of autoclave. There are 5 types of autoclave. They are gravity displacement autoclave, pre-vacuum autoclave, positive pressure displacement autoclave, benchtop autoclave, and horizontal or vertical autoclave. The first type of autoclave is gravity displacement autoclave. In gravity displacement autoclave, steam enters the chamber and displaces air, pushing it out through a valve. It is commonly used for sterilizing liquids and solid materials. The second type is pre-vacuum autoclave. It is a type of autoclave. In this autoclave, a vacuum pump removes air from the chamber before the steam is introduced, which increases the steam penetration. It is used for sterilizing porous materials and surgical instruments. The third type of autoclave is positive pressure displacement autoclave. In this positive pressure displacement autoclave, steam is forced into the chamber, increasing pressure. Air is displaced from the bottom of the chamber. The fourth type of autoclave is benchtop autoclave. This benchtop autoclave is a smaller units used in labs for sterilizing small volumes of liquids, culture media, or glassware. The fifth and final type is horizontal or vertical autoclave. This horizontal or vertical autoclave refers to the orientation of the autoclave door and its design, either horizontal or vertical. Typically, this type of autoclave is used in large industries or hospital settings. Instrumentation of autoclave. An instrumentation of autoclave contains 7 major parts. They are chamber, steam generator, pressure and temperature gauges, pressure relief valve, vacuum system, control system, and exhaust valve. Chamber. Chamber is the inner space where materials are placed for sterilization. Steam generator. The steam generator produces the steam required for sterilization. Pressure and temperature gauges. This temperature and pressure gauges are used to monitor internal pressure and temperature. Pressure relief valve. This pressure relief valve ensures that the pressure within the chamber does not exceed the safe limits. Vacuum system. Vacuum system is used for autoclaves with a vacuum cycle. For example, pre-vacuum autoclaves aiding in air removal. Control system. The control system is typically digital, allowing operators to set the sterilization time, temperature, and pressure. Finally, exhaust valve. The exhaust valve allows steam to exit the chamber after sterilization is complete, often cooling the materials down. Steps for using autoclave. There are 7 steps. The first step is loading. Place items in the autoclave chamber, ensuring proper arrangement to allow steam penetration. The second step is setting the parameters. Set the appropriate temperatures, pressure and time. Common settings are 121 degrees Celsius at 15 psi for 15 to 20 minutes. The third step is air removal. In pre-vacuum autoclaves, air is removed before steam is introduced. The fourth step is sterilization cycle. The autoclave builds pressure and temperature until it reaches the preset levels. This is maintained for the required duration. The fifth step is exhaust phase. Steam is vented from the chamber and the pressure is returned to ambient. The sixth step is cooling. Allow the contents to cool down before opening the chamber to prevent burns or contamination. Finally, the step 7 is unloading. Finally, remove the sterilized materials. Applications of autoclave. In medical and surgical instruments, autoclave is used for sterilization of reusable instruments such as scalpels, forceps, and endoscopes. In microbiology media, autoclave is used for sterilization of culture media, agar, and broth to prevent contamination. In laboratory glasswares, autoclave is used for sterilization of glass containers, beakers, and pipettes. In biological waste, autoclave is used for safe disposal of biological materials, including contaminated cultures or sharps. In pharmaceuticals, autoclave is used for the sterilization of certain drugs, syringes, and intravenous equipment. In veterinary practices, autoclave is used for sterilization of surgical tools and animal care equipments. In food processing, autoclave is used for sterilizing food containers and packaging materials. Advantages of autoclave. The advantages of autoclave includes highly effective sterilization, versatile use, non-toxic process, penetrates various materials, cost effective, standardized and reliable. Limitations of autoclave. The limitations of autoclave includes not suitable for heat sensitive materials, moisture sensitive items, proper loading required, limited to sterilizing certain materials, potential corrosion, energy and time consumption, risk of burns. Precautions for using autoclave. Autoclaves should not be used to sterilize waterproof or water resistant substances like oil or powders. The autoclave should not be overcrowded and the materials should be loaded in a way that ensures sufficient penetration of articles by the steam. The items to be autoclaved should always be placed in a secondary container. Only autoclavable bags are to be used to autoclave packaged waste. To ensure sufficient penetration, articles should be wrapped in something that allows penetration by steam, and materials like aluminum foils should not be used. The items placed inside the chamber should not touch the sides or top of the chamber. The wastes and clean items should be autoclaved separately. Attempts to open the lid when the autoclave is working should never be made. Liquid components should never be autoclaved in sealed containers. The liquid inside the containers should only be filled 2/3 of the total volume to prevent the spilling of the liquid. Plastic or polyethylene trays or containers should not be used as they might melt and damage the autoclave. Besides, never autoclave flammable, reactive, corrosive, toxic or radioactive materials, household bleach or paraffin embedded tissues. The paper should not be placed directly inside an autoclave as it is a combustible substance. It should be autoclaved in a waste bag on a biobag setting to prevent fire. Dear viewers, that's all about an autoclave. Thank you for the support. Thank you.